Hello, friends. Welcome to the second edition of Elets Diagnostic Leadership Summit. The theme of this summit is creating a future ready diagnostic sector. I am Renmoy Bhattacharjee, Assistant Editor with Elets Technomedia Private Limited, and I'm your host for today and tomorrow. It's my honor to welcome Dr. Li Yanfeng, Head of Business Development for IVD at Premise Life Sciences, who will present an industry presentation at this session. Dr. Lee is well trained in cellular biology and immunology with extensive research experience in signaling pathways determining B cell biology and T cell functionality. She authored in more than 10 world renowned journals and led over 50 I beg your pardon, 50 successful clinical trials connecting insights from customer behavior, skin biology, and intervention efficacy. Please welcome, with a round of applause, Dr. Li Yangfeng for her in depth and insightful presentation. Dr. Li, over to you.
Many thanks, Mimoy, for this exciting introduction. My name is Yanfeng. So as Mimoy has suggested, today I'll be really honored to present on behalf of Primas, our local partner, and I actually work for GenScript. So we are the manufacturer of this kit, and I hope that many of these people in the audience actually, have heard about our product, which is exciting invention. It's a neutralizing antibody detection kit, for COVID-19 vaccination monitoring. So let me explain the agenda. So the first few minutes, I'll try to introduce the SVNT technology. SVNT stands for the surrogate virus neutralization test. And of course, I'll actually follow up on the significance of the SVNT technology in the areas of correlate of protection for vaccine development and also potential and many popular applications of scientists using the SVNT technology to actually understand our own protection against the variants of concerns. So these are the available technologies for COVID detection at the moment. As we all know that the COVID detection during the acute infection stages are still being very popular. Widely used is the qRT-PCR or the rapid antigen test. However, for the post-infection serology or vaccination purposes, we do know that there's a lot of in-market kits that can offer the detection of binding antibodies, which is IgG, IgM, while you actually detects these antibodies that is induced by immune response to the virus or the vaccination. One last capacity in the world has actually been using is the virus neutralization test. Here, as you can see that the virus neutralization test can measure the functional antibody response or the immunity. So that tells you how much of protection you have after you've been exposed to virus or you have been vaccinated. So in that sense, I think especially today, as majority of the population is now receiving vaccination, it's very critical to understand our current immunity status after whether we actually are protected to potential uh, infection, especially when the delta variant and other variants is actually prevalent. So the innovation of surrogate virus neutralization test is actually dependent on the mechanism of how the SARS-CoV-2 virus enters our cells. So as you can see, the virus and cellular interaction depend on the two protein interaction. While you have the spike protein, and on top of it, at the top of this tip, is actually called RBD domain. And the RBD are supposed to bind to the receptor, the ACE2 receptor, to allow this virus to enter our cell. So the traditional virus neutralization tests actually rely on the use of live virus and also live culture cells. As you can see, such a process is very tedious. And what we have done is actually come up with a blocking ELISA technology. We, instead of using a live virus, we use this RBD in vitro express protein, and also the ELISA plate will replace what we currently use in the live virus assay, the human cells culture. We're using the SE2 overexpressed protein, which is already plated on the ELISA plate. So this is allowing a very simple surrogate virus neutralization test working principle. In the presence of neutralizing antibodies, the RBDs will actually be blocked and washed away. However, if there's no presence of neutralizing antibody, you will see a very strong light signal. So the samples with more neutralizing antibodies will show a lower signal intensity. The significance of measure the neutralizing antibodies has been documented in many places. So for the vaccine efficacy evaluation, based on the guidelines from the development and licensure of vaccines to prevent COVID-19, as you can see, there's always three arms of immunity. Of course, we look at the cellular response, which is majority in T cells, and then we have evaluation on the humoral response, the binding antibody test that I've also earlier on introduced. Most importantly, we wanted to see whether such vaccines, majority of these actually using the RBD technology, the purpose of it is to induce enough functional immune response, which is the neutralizing antibodies. So guess the neutralizing antibody has been also documented in the guidelines for investigational COVID-19 convalescent plasma. So basically, by manufacturing such convalescent plasma for the therapeutic purposes, we FDA actually recommended to go for the neutralizing antibody titer of at least more than 1 in 160. So such dimensions actually allow us to screen for those convalescent patients samples while they have strong neutralizing antibodies, so that this can actually help save more lives. And in the other pillar, we also recognize that majority of the current approved treatment of COVID-19 actually constitutes of different neutralizing antibody cocktails, or even there's many more companies are still in the development of new broad spectrum neutralizing antibodies. So how are we going to see whether one individual, after he's infected and he recovered, right? Do he still have the antibody, or is he protected at the days to come? So in this study, we actually have done a follow-up study for a total of 200 individuals. And you can see that among this cohort, we can see individuals who never have any neutralizing antibody at all. So this is a negative cohort, that they are just non-responsive. They don't generate any neutralizing antibodies. Three predominant groups actually contain individuals who have rapid waning. So these individuals can generate neutralizing antibodies, but by the time those antibodies will degrade, to certain very fast in this aspect. So by 6 months of time, the neutralizing antibody are already completely gone. Of course, we have luckier ones who sit in the slow waning group. So these individuals will still decline. However, they will still have certain protection in their bodies. And luckiest among this whole cohort is actually this persistent group, right? So basically, they started high, and their neutralizing antibody can actually last long as well. So this is a cohort following natural infection. However, we do see that, and also as many papers are also demonstrating that the vaccine-induced neutralizing antibody response following a very similar pattern compared with this. So how the neutralizing antibody actually have been uh, more and more commonly recognized as the best biomarker for correlate of protection. I would like to show you a few examples. So this is a very early paper done in last year, 2020. As you can see, there's a cohort of individuals who set out in the sea, and among this cohort, there are 6 individuals that are very interest. So basically, as you can see, 3 of them had positive binding antibodies, and when there is an outbreak on the boat, actually these individuals become um, susceptible. While there are certain individuals who actually show this was done by using our SVNT technology, who has shown positive presence of neutralizing antibodies, and these individuals were actually fully protected from reinfection. So the paper actually indicates that this is a very early indication that the neutralizing antibody from prior infection can significantly associate with protection, and it can protect individuals from reinfection. So after this, I show you a few more that actually when big data was generated by, for example, this cohort was done summarizing all the different vaccine platforms. This research was performed by a caliber scientist from the Australia. And when they look at the many manufacturer reported clinical trial results, right, based on Moderna, Novavax, and all the way to Sinovac, and what they have used was a matrix of neutralization titer compared to the convalescent plasma that was done in the same study, and on the protective efficacy that has been reported in such studies. Guess what? The correlation between the neutralization titer and protective efficacy has been very much significant. The higher the neutralizing titer and the higher reported protection efficacy has been seen. So this gives us a lot of confidence that after our own vaccine regimen, we do wanted to see that high neutralizing antibodies, and do take that, in our SVNT technology, it offers a very convenient way to measure the neutralizing antibody titer. As I just shown, the decay of some individuals can be very fast, even for neutralizing antibody titers. This paper also indicate that, the decay of neutralizing antibodies are also as rapid as the vaccinated cohort versus the convalescent subject, which means that as we are now see, many countries are already deploy a third vaccine jab to actually to alert the immune system to be reactivated, so that at least we still have that level of neutralizing antibodies for us to fight the existing prevalent variants of concerns. So there are the different platforms of the vaccines that are being incorporated in this Nature Medicine paper, and I would just like to know, to share that no matter the traditional or the novel vaccine approaches, the key indication here is actually the higher neutralization antibody titers, and that will give you the highest efficacy of protection. And the recent paper that has been published in Israel is a very good live example. So Israel has been very comprehensively protected by the Pfizer vaccine, and the healthcare workers actually had the 2 cohorts within the study. That one is called the vaccine breakthrough group. So these individuals actually, you can see that they have lower amount of neutralizing antibodies. However, in the same study, in the same healthcare, premiums, when individuals who are more protected against the infection was actually having much higher neutralizing antibodies. So in this report, actually, we also use the international unit to convert the neutralization antibodies to cPass the SVNT technology results, and we can safely say that in the respect of the the different variants of concerns, cPass SVNT technology reported 80% and above is actually a good threshold for us to identify individuals who are more protected. And individuals lower than this threshold, either you watch carefully and really maintain your social distance, but these individuals may need to go for a booster to indicate that to allow your immune system to respond to actually give you the necessary protection. So with that, I would like to share with you, right, in the Israel real life efficacy data, within just 1 month's time difference, we see the vaccine efficacy dropped from 94% to 64%. Why? Everybody knows, right? Because we have all the variants of concern circulating now in the society. So the SVNT technology is a powerful technology to actually allow us to differentiate the neutralization capacity of our sample, being a patient sample, or actually later on I will address the vaccine samples as well to see how much of a protection of neutralizing antibody you may have against the different variants of concerns. So the study data shown here, right, that the wild type and also the alpha strain have very similar neutralization capacity. However, individuals who are infected by the wild type strain actually generated very little protection against the beta variant. And also for the same individual who actually infected with the UK variant, these individuals were also not so well protected against the beta, right? So vice versa. We have done this same study in the USA as well, and a similar trend was actually observed. So this is more of interest to individuals who are not um, infected but who actually received their vaccine regimen. So this study was actually performed in the USA, as the study actually have collected sample from the different vaccine platforms, Pfizer, Moderna, and Johnson Johnson, and of course there's the control. So as you can see that when we look at the SVNT results of individuals who actually have received Pfizer vaccine, their protection against the original virus, of course, the highest, and then the beta alpha is slightly lower. And do take note that when you look at the Johnson and Johnson, you do have some level of protection, 56% when reported by SVNT. However, that protection against the variants of concerns very much dropped, right? So we do see the same phenomena in the study of Singapore cohort. And what we now understand is that so the neutralizing antibody is very critical. It has been also shown by WHO committee of experts in the last committee meeting to show that it can be a good biomarker to indicate how the new vaccine generation and development should be referred to. So we do have our collaborator here having a fantastic publication who used SVNT technology and compare, what kind of vaccine regimen can be the best cross neutralizing antibody generation. So in this cohort study, right, it was a follow-up study by looking at the SARS-CoV-1 patient. These patients were actually sufferers of SARS in 2003, right? 17 years later, these individuals have been received their vaccine of Pfizer vaccine in Singapore. So as you can see, the SARS-CoV-1 patient itself does not have any much neutralizing antibodies against the SARS-CoV-2 and the variants of concerns. However, the SARS-CoV-2 patients do have, significant amount of neutralizing antibodies against the variants of SARS-CoV-2 viruses. And that actually included all the way to pangolin bat. So these are actually tobacco viruses. It's a family of, it can be even inclusive of all the new variants of concerns that are yet to come. Right, healthy vaccinated, very good protection, and we do see some reduction in the protection against the beta. Right, we have also seen this reduction in many other reports as well. So the SARS-CoV-2, and then they're vaccinated, and their protection against the Sabbatical families are very much reduced. Look at the last cohort. So these SARS patients, when they recovered 17 years ago, and then received vaccination of Pfizer 2 doses, right? As you can see that their neutralization capacity has been fantastic. So basically, by recovered patients receiving SARS-CoV-2, your immune system can actually be triggered in a way to produce a very broad spectrum of neutralization capacity to not only protect you against SARS-CoV-2 original virus, but the mutant variants of concerns, and all the way to the other Sabbatical families that can originated from pangolin bat, different viruses that they can share, a very huge variability in the sequences. So for this study, it actually triggers a very innovative way of designing the next generation of the vaccines. So basically, this group of one is actually now looking at how we can generate the next generation of vaccine, strategy by combining the different sequences between SARS-CoV-1 and SARS-CoV-2, and see how we are able to generate a vaccine that is able to protect us uh, against the variants of concerns that may emerge in the next days and months and years to come. Basically, how the SVNT can be utilized to differentiate neutralization capacity to the variants of concerns is very simple. So basically, we can make use of the current existing kit, and then whichever variants of concerns that team may be interested to evaluate, we can actually add one additional SRBD HRP labeled proteins. So this actually works very beautifully for us to compare our vaccine or infected cohorts, their neutralization capacity against the different original virus or whichever variants of concerns that the researcher and scientists may be interested in. So do feel free to reach to Prima's team to actually get more information. How can you get access to this platform? With that, right, we all know that for binding, it's very difficult to actually allow us to differentiate between the variants of concerns because the binding antibodies doesn't really, have that capacity to differentiate. In with that, I would say that I really appreciate Tin to spend the quality time with me, and I would like to, if you have any questions to follow up, interested in this technology, to speak with our expert product manager, Mr. Manny, and he will be there for you. Thank you so much.
Thank you, Dr. Li Yanfeng, Head of Business Development for IVD at GenScript Asia Pacific, for speaking about neutralizing antibody detection for COVID-19 post-vaccination monitoring. That was a wonderful presentation. You brought out so many relevant and recent papers from around the world. Thank you so much. And as a token of our appreciation, we'd like to present you the participation certificate. And if I may request my team to bring it up. Thank you, Dr. Lee, for joining
Thank you.
this Diagnostics Leadership Summit brought to you by Elets. Thank you.
Thank you so much, Nimoy. My pleasure to speak to you.